Hello, my name's Jake Bradford. I am a PhD candidate at the Queensland University of Technology in Australia. Thank you for connecting into my poster presentation on my work in developing Crackling, which is a fast and accurate method for designing CRISPR guide RNA sequences. I would like to acknowledge my colleagues involved in this work, Dr. Timothy Chappell, and my PhD supervisor, Dr. Dimitri Perrin. CRISPR-Cas9 is the gene editing tool that has revolutionized a geneticist toolbox. CRISPR is a genomic region and is essentially a memory bank of past viral infection. It contains snippets of viral DNA, which are each separated by the same short and palindromic sequence. Alongside this, a very interesting set of CRISPR-associated genes, or Cas genes, can be found. This is where you'll find Cas9, for which in wild-type systems is a nuclease capable of cleaving viral DNA. Cas9 is directed to cut DNA by a guide RNA sequence, which has previously been obtained and stored within the CRISPR array. However, in the lab, Cas9 can be used with the synthetic guide RNA sequence to target any gene on any genome. Cas9 is easy and is cheap to use, though support from bioinformaticians is needed due to some challenges around designing this guide sequence. For this, a number of pipelines exist. In some previous work, we reviewed and benchmarked these existing pipelines and described what a high-quality pipeline should look like. Any decent pipeline should consider two properties: guide efficiency, that is, how well the guide will bind at the desired target site, and specificity, that is, how unique the guide is to the target site. These are two complex properties. For efficiency, some methods follow the pattern of using established biological rules, while some others consider machine-learned models. We found that both approaches lack value and therefore developed a new approach, which I will discuss soon. For specificity, some use rudimentary methods, for example, a sequential search, where every guide is compared with every potential off-target site. Some others use complex data structures for fast off-target lookup, though sometimes these do not scale well. Overall, we found that most pipelines don't provide a lot of value and are slow or just can't perform on large-sized genomes. Therefore, we developed Crackling. Our goal was to produce Crackling to be more precise and fast in order to analyze large-sized genomes in a reasonable time. When evaluating guide specificity, it's important to consider other sites that differ by up to 4 mismatches when compared to the guide sequence, as Cas9 may cleave at these positions. This becomes a computationally expensive task. In previous work, we found that this often causes speed bottlenecks. To overcome this, we utilize an approach named Inverted Signature Slice Lists, or ISSL. ISSL constructs an index of off-target sites for any genome and is able to evaluate guide specificity up to an order of magnitude faster than others. We tested this across 12 genomes on 2 machines, with repeat tests to find an average result. The smallest genome was of size 374 megabases, and the largest being nearly 10 gigabases. Figure 1 in the poster shows the results of some existing tools. Find mismatches and find mismatches bit are the same, just with bit encoding being utilized in the latter method. Here you can see that find mismatches bit performs an order of magnitude faster than its original version, and is performing about the same compared to Flash Fry and CRISPR. Then, another order of magnitude faster, we see our new approach based on ISSL. It was able to complete all tests on all genomes at a much faster rate than others. But just because we're quick doesn't mean we're accurate. Let's talk about why Crackling is more precise than others. Last year at ISMB, we presented a new approach for selecting highly efficient guides. In this approach, we take the rules implemented in the existing 3 most precise pipelines and only accept a guide if at least 2 of these agree that it will be efficient. We tested this approach on 2 datasets, which contain experimentally validated guides. We found that the consensus approach was more precise than any tool taken individually. Figure 2 shows this. In panel A, a blue dot indicates that Crackling has predicted that the guide is going to be efficient. Ideally, we want Crackling to be predicting this only for those points appearing in the efficient region, as these guides cause most gene knockouts in the experimental study. Considering every blue point, 86% of them appear in the efficient region. That is, Crackling is correct 86% of the time. We found a similar result on the second experimental dataset, which results are shown for in panel B. A large majority of the efficient classifications appear above the threshold of 0.8, which indicates the guide is experimentally efficient. Overall, the consensus approach provides more precision than any other pipeline when considered individually. Combining both the consensus approach and ISSL, Crackling is precise and very fast. As you can see in Figure 3, where Crackling is a cyan line with square markers, it is amongst the fastest performing tools when tested using our previously published benchmarking method. If you're interested in knowing more about how the ISSL approach works, Figure 4 and Figure 5 provide more detail. You can download Crackling via the GitHub repository, which includes setup instructions. We also have a paper on bioRxiv that provides more information. I hope that you have enjoyed this presentation, and thank you for your time.